as we continue, we looked at the synthesis of nitrogen-containing compounds, and we started with actually fixing nitrogen, meaning breaking the nitrogen bond, the triple bond, into ammonia, which then in biological systems in the buffering system become the ammonium ion. And then we're going to incorporate them into amino acids. How many amino acids have we synthesized as of this moment in time? 2. Glutamate, the first one that we incorporate the amine in, and then glutamine. Now, glutamate and glutamine, that interaction is really important. Glutamine particularly is a wonderful substrate for a lot of different compounds that contain nitrogen. And what we can see is the regulation of the enzyme that converts glutamate into glutamine is a little bit controlled, okay? And so we have lots and lots of feedback. Every one of these outcomes, AMP, tryptophan, carbamylyl phosphate, glucosamine 6, histidine, CTP, glycine, alanine, are all allosteric inhibitors to this process. Now, this tells me a couple things. That this particular intermediate glutamine is a precursor to a lot of different types of compounds. Secondly, this enzyme is probably going to be at least functional at some level all the time. This particular enzyme is neither 100% on or 100% off. And so it's modulated by how many things and what the concentration of each one of those is in the body at any one time. Also, some of these are mutually exclusive, meaning that you can't actually have all of these concentrations high in one cell because they are actually balancing two different types of processes. So you are going to have a very controlled level of activity that can span all the way up between 0 and 100%, and most likely it is running. Now, AMP, energy generation, CTP is also a function of energy generation or the nucleotides synthesis. Tryptophan, histidine, glycine, and alanine are feedback from synthesis of proteins. Carbamylyl phosphate, where did we see that particular molecule? What pathway? Carbamylyl phosphate is part of the urea cycle, okay? And so that is the degradation of amino acids. And so if that goes up into concentration, we have slowing down of this reaction. And then glucosamine is a function of amines being incorporated into the sugars that are mainly on the outside of cells for cell signaling. And so there's carbohydrates in there, there is amino acids in there, there is the breakdown of amino acids that are being signaled, there are nucleotides. It's very, very controlled. Now, in the mammalian system, can we synthesize every one of the 20 amino acids? No. And so there are what are called essential and non-essential amino acids. And so the essential amino acids are the ones that we must eat so that we have the amino acids available for synthesis. Non-essential means those are the ones that we can synthesize internally. There are a couple caveats. If you actually looked in the urea cycle, we do have a reaction in enzymes that make arginine in that particular pathway. And so why in the world do we have to eat arginine if we know we can make it? Well, the arginine that is made within the the urea cycle, it gets converted into urea itself and keeps the cycle running. And so we cannot draw from that arginine, or we will stop the urea cycle. If we stop the urea cycle, we'll become toxic in terms of the ammonium content in the body. So we have to eat arginine for protein synthesis. Tyrosine, on the other hand, we do not have the enzymes necessary to synthesize tyrosine completely. But what we can do if we eat phenylalanine, which is a essential amino acid, as part of the breakdown products we saw, we do have the enzyme to convert phenylalanine into tyrosine. And so we can do one step and create tyrosine, but we couldn't synthesize tyrosine in its totality if we're absent phenylalanine. So this looks like a wonderful multiple choice question. Which ones are essential or which ones are non-essential? All right. Non-essential amino acids are synthesized from the citric acid cycle, glycolysis, pentose phosphate pathway intermediates. And every time we say glycolysis, we could also mean gluconeogenesis. There are only 6 biosynthetic pathways that are active in the mammalian system. And so let's start to synthesize a few of these particular amino acids. All righty. Who wants to be the first volunteer, or do you want me to start choosing? Look at the slide, take a look, see what the reaction's going on, and when you feel comfortable, choose. Someone jump up here. No one feels, oh good, we got somebody who feels comfortable. All right, in every reaction, we always have a base and an acid to react. A lot of times we actually use a base as the initiator, but this one is already deprotonated. And so you're going to lock the double bond. Electrons will attack the phosphorus, kick up, kick down, and ADP is the leaving group. All right, perfect. Now you get to choose someone else to come up here. Okay, what kind of enzyme does this reaction? I didn't give you the name, but tell me what class of enzyme. You start with glutamate and you have a phosphorylated intermediate at the end. It's a kinase. Okay, come on up. All right. Let's change, that yellow was kind of, you couldn't see. Oh, I'm going to do something for you too. Instead of NADPH just written like that, I'm going to write NADP with a bond in H. Okay. And so the hydride is able to attack, kick up, kick down, and the bond between the carbonyl and the oxygen, the electrons go into the oxygen. Very good. Choose someone else. And the leaving group is phosphate. All right. Can you tell me what class of enzyme this would be? A reductase, an oxoreductase, most of the time named a dehydrogenase. Or the NADPH allows us to use a hydride as a reacting group. All right? And so you didn't take Biochem 2, so he is ineligible. All right? So who would you like to choose? Now you kind of feel like a professor. Anybody's got to choose, put the head down, right? All right. Is it Jose? Come on up, you get it. I'm sorry. Oh. There's social consequences with this, isn't there? Okay. All right, remember that we always have an acid and a base, and I will put the base nowhere in particular and the acid nowhere in particular on the slide. All right, you're going from this starting material into the intermediate stage, and we are making proline. So if you take a look at the beginning and the end, we'll do some inventory. We see the acid there. That didn't change, right? That hydrogen is still there. But this nitrogen right here has how many protons?
Has 3.
3 and 2 there. So that kind of gives us, we're going to have to take off a hydrogen.
Which one we attack?
No, it doesn't matter which one we attack because all 3 are equivalent.
Okay.
So just now it's your convenience. Okay, now that pair of electrons are negatively charged. That can attack something.
Okay, so would it go to the...
It's got a carbon there that has an oxygen attached to it. Where's the carbon in here that has an oxygen attached? Mm-hmm.
So this hydrogen would attack the...
Always start with the electrons. So the electrons are going to attack, and if it attacks at this bond, that's partially positive because it's a carbonyl. So this negative pair of electrons can do that.
So it does attack from here to here?
Mm-hmm. And that attack creates that ring structure. Now, too many bonds to carbon, so the double bond does what?
It would kick up, and then it would pick up that hydrogen.
Mm-hmm. And then you can show, yep. And there you go. All right. Go ahead and choose someone else to learn. Now, that ring closure starts to look a little bit more like proline, okay? Because we know we need to get a intermediate that has a 5-membered ring. And so who is the next learner? I know it's so easy when you're sitting in the seat and so hard when you get up here. It's been a really long time. Okay, so you're always going to have an acid and you're always going to have a base to use. So I'm going to go ahead and just nowhere in particular give you a base and nowhere in particular give you a proton, okay? And so when we look at the beginning and the end, you see places that really don't change anything, so we're not going to do anything. But that nitrogen has 1, right?
Yeah.
This nitrogen has 2. And then this hydroxide is missing. But look what comes off. Water.
Okay. So, is it going to attack hydrogen here?
So the pair of electrons gets it. Good. Now, that pair of electrons, where does it end up? It was between the hydrogen and the nitrogen, but look at the product. Those pair of electrons.
The double bond.
Correct. So just, yep, perfect. And then what leaves?
The H2O.
Yes, and so the electrons between that carbon and that oxygen will go grab a proton to become water. And so,
Oh, it's over there already, sorry.
that's okay. There you go. And you have done it perfectly. All right. We have closed the ring. We are not at the amino acid yet, but we're very close. All right, and we went through an intermediate. Okay. The last reaction, go ahead and volunteer someone. Here's another dehydrogenase. Whenever we see NADPH, that means we invoke a hydride, can be a nucleophile. And we spend a lot of time using glucose to make NADPH so that we can have the reductive power.
So hydride attack here.
Okay. If it detects there, then what happens?
It's double bond. Let's pick up that.
Yes, that is one of the possibilities that could happen. I'll show you another possibility. Thank you. And if you actually have to prove which one it is, we have to actually do some labeling. But you could do this. You could attack there, okay? And then the double bond again will then pick up the proton. All right. We've got 2 different options. The first, the attack on nitrogen, and the second is attack on carbon. If you attacked here, the electrons would temporarily need to be here, okay? And that's a carbanion. If I attacked here, then the electrons temporarily go into the nitrogen, and then it would be neutral. All right. Organically, this one is a little bit more stable, okay, because the intermediate form is neutral, whereas if the blue, it would be negatively charged. Now, the enzyme could have a intermediate that's negatively charged. Yes?
Thought the blue one made it neutral on the nitrogen.
Nope. The blue one, when it attacks, it makes the nitrogen neutral, but it makes the carbon negative. Okay? When the, what is that, yellow? Okay, good. Yellow attacks, colorblind professor using colors, not good. Okay, yellow attacks, the carbon is neutral and the nitrogen becomes neutral in the intermediate form. Now, on an exam, I would have accepted both of those because the enzyme could have stabilized either one of those attacks. All right, and we have created proline. We have 3 of 20. All right, serine. Okay, who is next? Did you choose? Did you do your other job? Oh, she knew it was coming. I could see it in her face. Okay. All right, here is a dehydrogenase. All right, because I'm going to have an NAD plus, and that allows a hydride to leave. Also, we start off with 3-phosphoglycerate. What pathway are we starting with the starting material? You don't have to answer. Which one? Glycolysis. That is half right. Was the other half of it? Or gluconeogenesis. Okay. I gave you flashbacks. Oh, no. All right, I'm going to do a couple things for you. All right. I am going to draw out and articulate that hydrogen out there. Okay, we always have what in a reaction? A base. We always have a base to use. I'm going to put it somewhere randomly on the screen. Okay, and so now as we look, do you see what changes? Mm-hmm. Yes, and then something has to leave. Yes, and where does it go?
I'm gonna say NAD.
Yes, NAD+. And that's the only reason why we could allow the hydride to leave is because we have an NAD+. All right, perfect. Go ahead and select someone else. All right. And so we've done that particular oxidation. Okay, yes?
On an exam, would you have the structures?
On the exam,
You would just add
correct. What I would have on the exam is written this structure, and these intermediates. The only thing I will not draw for you is a general base and a general acid because you always know you have that available. Okay? Yes?
And would you possibly ask us what class of enzyme?
Would I possibly ask what class of enzyme? That seems like a good suggestion for an exam question. I'll consider that. Okay? All right. Next person coming up. Who is okay? I'll let you sit. I'll let you sit for just a second. All right. This particular class of reaction, all right, something called an alpha keto acid, okay, and then I transfer an amine on there. This type of reaction is a very complex reaction. We have seen this in the degradation of amino acids. It uses a cofactor to get this done called PLP, and this is called, or this type of reaction is a transaminase. It is a multiple step, I think it takes like 10 different steps through using the cofactor to do this reaction. I am not going to ask you to do that and write that particular mechanism out. I even made you do the quiz where you had all the resources to get that done. But recognizing that a ketone is converted into an amine is a big class of reactions that is converted by the transaminase. All right, come on down. All right, we've got 3-phosphoserine, and we're going to make serine. We're about ready to make our fourth. Okay, I'm going to do one thing. I'm going to lock the double bond, and I'm going to have a negative charge and a negative charge. All right, you always have a base, okay, to attack. And there are two acceptable answers on this mechanism, okay? So, what do you see is going on here? You can't attack there, yes. What is going to attack there? Okay, so a base usually picks up a proton, so what are you taking with the base with that? Bases usually pull out a proton. Was there a proton there? Okay, so nothing happened. Okay, what is the other reactant? Yes. Okay. If you take the proton off of water, oh, you need to attack a proton. That's usually what we take. Okay, good. So we got the proton. All right. That means the pair of electrons between the hydrogen and the oxygen are now negatively charged. They can act as a nucleophile, and they can attack something. Yes. Okay. Now, every time you attack something that has a double bond, what has to happen? Correct. Kick up. Good. Kick down. Great. Now, a bond has to fall off. Now, you always have a hydrogen, so go ahead, proton, go ahead and put it there. Okay. And so, as you put that down, I have to break a bond. So the electron pair between the phosphorus and the oxygen. Yes. And that will be the proton that is on the hydroxide. So that last movement puts this proton onto that oxygen and becomes this hydroxide. The only thing I would clarify is that that pair of electrons came and did it. Because you took, this first one took that proton, and then this pair of electrons is what attacked that phosphorus. Perfect. All right, serine's been synthesized. So how many now do we got? 4 of 20. All right. Now the aromatic amino acid synthesis. They are essential amino acids. That means we can't synthesize them, but there is a pathway in plants and microorganisms to do that. We're going to take a look at the synthesis of phenylalanine, tyrosine, and tryptophan, and we're going to look at them in the enzyme E. coli, and we're going to see that they are interconnected. So we're going to create some molecules by which they are, from that one molecule, go to all the rest. And so if any synthetic pathway has a common theme, we need to know what that common structure is from which they are synthesized from. Oh my goodness gracious, great balls of fire. All right, this first particular reaction, I've got urethose 4-phosphate. What pathway? The pentose phosphate pathway, and that's actually in the regeneration steps, the non-oxidative portion. And then phosphoenol pyruvate comes from glycolysis or gluconeogenesis. And so knowing where these starting materials come from is probably one of the very important things that I want you to understand. And so we're drawing from 2 different pathways to synthesize this. Now, urethose 4-phosphate and phosphoenol pyruvate can be created from glucose itself. Okay, and so I can take 2 glucose molecules, end up with these 2 intermediates, and we can, in the plant, start to synthesize what's going on. All right. In with a partner or two, look at this reaction and tell me what connects to what. Okay, which two carbons connect together to get this the beginning of this ring structure? Okay, work with your neighbor and decide. This is a little bit more complex than anything we've seen up to this point in time. So, what am I going to do? Well, this is what I would start doing. I'm going to call this one 1. I'm going to call this one 2. I'm going to call this one 3. And then I'm going to come over here. I'm going to call this one A, B, C, D, and E. Okay. Now, where in the world? Okay. There is one acid on this side, right? Okay. How many acids are on this side? Acid groups? Just one, right? So I can say that one is 1. If that one is 1, what is this one? 2. And what is this one? 3. Okay. All right. That's kind of cool. All right. On this side right here, there is one methylene, CH2. Where's that CH2 over here? Right there. So this one must be the D. That must mean this is the E. So this one must be the C. This one must be the next one must be the B. And this one must be the A. Okay? So you can see that the carbon 3 and the A must be put together. Now, there's something different about A, isn't there? A here is a what? Carbonyl, right? And A here is a hydroxide. All right. What else happens around carbon 2 here in the product versus the reactant? Yeah, there's a double bond, and there is no phosphate. All right. So, what in the world is going on here? Okay. Here's what is going to happen. I always have a base, correct? I am going to take a proton from water. I will then attack the phosphate, because phosphate has to leave because it's not in the product. Every time I kick up, I kick down to form the double bond. And a lot of times, what we'll do is we'll take this pair of electrons and throw it right onto the oxygen. But that's not what happens in this case. Okay? Between this oxygen and that carbon in the product, what happens here? It becomes a what? Double bond. So instead of the electrons just going onto the oxygen, I'll actually form the double bond. Well, that's a booger, because I now have 5 bonds to carbon. But I have a double bond that will be able to attack, and by my inventory, I said I had to attack it to the carbon that was labeled A. And remember, I always have a proton, and then I pick up the proton. So in 1, 2, 3, 4, 5, 6, 7 easy steps, I form the product. Okay. Do you want this one on the exam? Well, now that you've seen it, but that was rather complicated. Okay, that one is more complicated than I would assume that would be one on the exam. if you wanted to be a really mean professor, this would be the first question on your exam. And then you would feel, I can't do anything. And it would be really, really bad. And so this is a little bit more complex than I would want you to really recognize in terms of an exam, but maybe dealing with this in a homework and attempting to try to reason it out. This would be more like a homework type of interaction. All right. In this, it's still not really easy. Okay, there are some intermediates here. And so to go from the intermediate that we've just made to the transition, we're going to always use a base. Okay, and in this case, I see a phosphate come off, and I see I'm going to take a proton off. So if I take a look at my inventory, nothing changes until I get right here. Okay, here I have a hydroxide, here I have hydroxide. Here I have a hydrogen, there, oh, I don't have a hydrogen. And then this is missing the phosphate. So I take that hydrogen, form the double bond, and phosphate leaves. All right. The next step, if I take a look and do some inventory, the acid, the carbon, hydroxide, the carbon, So I know there's going to be a reaction here. Come around the ring, no change, no change, no change, definitely a change. This oxygen is a hydroxide here and a ketone there. And most of the time I'm looking for where's the proton missing, and I found the one that is missing. I take this, I form the double bond. That allows these electrons to attack. I always have a proton, and I pick up. Now, if we're heading towards the aromatic synthesis, we have to have a ring structure. And so I have created my ring. Now, that doesn't look like any of the amino acids that we know that are aromatic yet, but we're going to get there. Okay, dihydroquinate. All right. In this, we're going to lose water. All right, and if I take a look, start here, no change, no change, no change, no change. There is a change. I lost a hydroxide, and over here, how many hydrogens are understood to be at this carbon here? 1. How many did it start with? 2. I found what I have to do. Take that proton, form a double bond, pick up water, or pick up a hydrogen to create water. 3-dehydroshikimate. I love it, shikimate. All right. I have a dehydrogenase because I have a redox reaction occurring. So I know I have a hydride that can attack, and as I look around the ring, the acid, no reaction, no change, no change, no change, definitely a change. Ketone into a hydroxide. The hydride, which is a negative nucleophile, will attack the carbon, which is an electrophile. And that gets converted into the alcohol. I now have shikimate. Sounds like some sort of something you order in a sushi bar. I don't know. Okay. Shikimate will then react to make the 3-phospho shikimate. And to do that, I'm going to go ahead and articulate this particular proton out. Base will take the proton. The electron pair will attack at the alpha, beta, or gamma. Gamma phosphate. Kick up, kick down, and the electron goes on to the oxygen. What class of enzyme does this reaction? A kinase. And we've got shikimate 3-phosphate. And now, shikimate 3-phosphate reacts with PEP. What is PEP? Phosphoenol pyruvate. What pathway are we taking this from? Glycolysis or gluconeogenesis. Okay, this is kind of a weird reaction. The enzyme is involved in this reaction, and so you will actually not be responsible for this particular mechanism. In essence, this hydroxide and this carbon react, and the phosphate will be the leaving group, but it is more complex than a simple deprotonation and attack. Then we get into naming that is really interesting, 5-enol pyruvyl shikimate 3-phosphate. Ooh. And in this particular case, we are going to dephosphorylate this, but this is kind of an interesting reaction. All right. Name for me this type of reaction right here. All right. Okay, and if I have a base, and it takes this, and that falls off to form, what type of mechanism did I draw? What would you label this as? An elimination, E, and this would be a 2. Okay. Okay, what kind of reaction is this? It's an elimination, and it's an E1 mechanism. Okay. All right. Let's take a look at this particular reaction. All right. We definitely have a change right here, correct? On this carbon, I have a phosphate. That same carbon over there has no phosphate, right? What else is different around that carbon? Yeah, this is a single bond, now it's a double bond, okay? Did I do anything here? No. Did I do anything here? No. Did I do anything here? Yes. Okay, how many hydrogens are understood to be there? 2. How many are understood to be right here? 1. Okay. Did I do anything there? And did I do anything there to that carbon? In terms of the number of atoms that are bound are exactly the same, just there's a different placement of the double bond. Okay. Now, watch this. If I have an extra base, if I have an extra hydrogen, I'm going to use a base to take that off. Okay. Now, watch what happens. This pair of electrons forms that double bond, right? Which then means there's too many bonds to carbon right there, right? So what I'm going to do is I'm going to make that bond flow down, and that then allows the phosphate to leave. Okay. This particular reaction is an elimination. Okay. It has two things involved, the base and the reactant. So it is also an E2 reaction, but this is called an E2 prime reaction. The reason is an E2 reaction up here at the top, the proton that is taken off is alpha to the leaving group. Okay? So I take this off, the electrons form the double bond, the leaving group. This one is not alpha to whatever leaves. It is actually connected to the leaving group through a double bond. And so this is a elimination that is 2, but the prime means that the elimination is connected through a pi bond movement. All right. I will, on the exam, draw mechanisms for you. And the ones that you're responsible for are the E1, E2, and now E2 prime. Also, the mechanisms of SN1 and SN2. So there are 5 different mechanism types that you should be able to distinguish. If I write out the entire mechanisms, you tell me which one of the 5 it is. All right? 4 of them you learned in organic, and then I reinforce them, and then this is a new one that you learn in biochem. All right? Corsmate. This is the intermediate that is common to all 3 aromatic ring systems. Okay? The branch point allows the corsmate to be the precursor to phenylalanine and tyrosine, and then a mutase will convert that, will take it in another direction when we actually do the reaction to get to the tryptophan. This mutase is kind of interesting. Again, it's more complex a reaction. I wouldn't assume that you would be able to do this, but in essence, what happens is that reacts, this reacts, that goes there. It is a cyclization, 3 electron movements. Okay. Does A or 2 or delta go first in the reaction? Which movement is first? Could be. But if 2 moved, then the delta movement had to happen, and if the delta movement had to happen, then A has to happen. Since it's cyclic, any one of them could have been the beginning move. It's actually more complex than that, but essentially that's where the ultimate movement actually occurs. All right, here would be one type of reaction that I could actually ask of you on the exam. And so I'm going to go down 2 different ways, one to make tyrosine, one to make phenylalanine. Really the only difference is, do I have a hydroxide, do I have a hydrogen in the para position of the ring structure. All right, one is a dehydrogenase, an oxidative decarboxylation. The other one is a decarboxylation with a hydroxide leaving group. And so anytime that we have a decarboxylation, we have an acid, and the acid will allow its electrons to form the double bond of the CO2. In this case, if I look at the final product, I see a double bond in that particular position in the reactant, in the product. And then this moves, and then something has to leave. On this side, the leaving group is the hydride. And that is because it has an electron acceptor. That isn't what you might expect, but only because the enzyme allows it. The other movement is probably what you might expect, where we have the hydroxide coming off, which can become water, and both are decarboxylations. We are very close to getting to our tyrosine and our phenylalanine.